All right, guys, ACA class. So this is our cellular respiration foldable. I just kind of want to review where we were. This is the drawing that we had of our mitochondria. We have the important structures labeled in red. There you go, get the whole thing in there. We also have, our important information, our structure of the mitochondria, how each of the four pieces of the structure is important, and specifically how they play into the importance of the mitochondria. We then went on to glycolysis, where we started with a glucose. We added some phosphates from ATP to make fructose 1,6-bisphosphate. We split that and made 2 G3Ps. We then transferred some electrons to an NAD plus to make NADH. We got rid of those phosphates so that we ended up with pyruvate or pyruvic acid, and we repeated that with the other G3P. We made a note up here of 2 ATP are needed to provide energy to start the reaction, but 4 are made, so we have a net gain of 2. We talked about our redox reactions, focusing on oxidation is losing electrons and reduction is gaining electrons. And then we introduced the concept of an electron carrier, which is a molecule that carries electrons, and in this case, it is NADH is carrying the electrons, and that is made with an NAD plus, 2 electrons, and a hydrogen ion. Then we went on to the citric acid cycle, where we start with that pyruvate that we made and the end of glycolysis. We add a coenzyme A, we remove a carbon dioxide. In that process, we lose some electrons, so they're picked up by the NADH. That goes into our cycle where our 2-carbon CoA molecule combines with our 4-carbon molecule. We lose the CoA, that's 6 carbon molecules. In the process of going from 6 carbons to 5 carbons, we're going to get rid of a carbon dioxide, and then we'll have some electrons released that are captured by NADH. We're going to do the same thing when we go from 5 carbons to 4 carbons. We're going to release a carbon dioxide, capture those released electrons with NADH. But here at the end, we're done. We have 4 carbons, but we need to get to this specific arrangement that actually looks more specific than 4 carbons in a row. I'm going to make some ATP. I'm going to release some electrons and capture them with FADH2, so that's just another electron carrier. And then I'm also going to capture some more electrons with an NADH. We talked about the fact that this happens in the mitochondrial matrix, that this is also called the Krebs cycle, okay? And we talked about the fact that carbon atoms are released as carbon dioxide. This FAD is an electron carrier. When it's FADH2, he is formed with an FAD, 2 electrons and 2 hydrogens, so that's FADH2. We also have a note that our starting molecule is pyruvate, and that we're going to make 1, 2, 3 carbon dioxides, 1, 2, 3, 4 NADHs, 1 ATP, and 1 FADH2. We did make a very important note that there are 2 cycles of the citric acid cycle are required for each glucose, because we made 2 pyruvates here in glycolysis. All right, so now we need to get started with our electron transport chain. So the electron transport chain is a little bit more complicated, okay? I'm going to use my picture. It's going to take up most of the space. It is made of a phospholipid bilayer. It happens in the inner and outer membrane. So I need a phospholipid bilayer. And I'm not going to want to draw all these circles. So after I draw for you, okay, this is my outer mitochondrial membrane. Okay. Then I'm going to have my inner membrane. All right, and I'm not actually going to draw this with pen. I'm going to use pencil. Because I'm a little bit of a perfectionist and I'm going to have to erase some of my markings, okay? So I'm just going to draw my lines across in pencil, okay? I'm going to label this as the inner. I'm going to label it here. Inner mitochondrial membrane. Okay, so that means that this space between the two membranes, if you remember, is called the intermembrane space. Or the space between the two membranes. All right, if you want to add some colors, that might help, okay? All right, so now I'm going to draw in my proteins that I'm going to need to reference. They are transmembrane proteins. Okay. All right, so these are all my proteins, so now I can erase my little lines. And now I can replace these with pen. All right, so now we are going to focus on what happens. I'm going to use my red. Just as we did on the first picture, because I want to make sure that we're focusing on what's happening. So we're going to start with our NADH, our electron carrier. That comes in, so this is our mitochondrial matrix. Let's actually label that. So that means that this NADH, it was just made from the Krebs cycle, or it was made in glycolysis, okay? And what's gonna happen is he is gonna break into his NAD+, his hydrogen ion, and release 2 electrons into this transmembrane protein complex 1. Okay, so all right, so that's the first thing that happens. This NAD plus that's released, that can then go on to back to the Krebs cycle and be used again to pick up more electrons. All right. These 2 electrons are going to be passed onto this little Q protein. This is a little guy. He actually starts out over here. He picks up these electrons and then he moves through this protein complex 2, and then he is going to transfer them to this cytochrome C little transfer protein. This little transfer protein is then going to move those electrons into protein complex number 4. Okay, we're going to hold on to them for just a moment before we do anything with them. Okay, when these electrons go from protein complex 1 to the Q protein complex, another reaction occurs. That other reaction is when these hydrogen ions are then pumped from the mitochondrial matrix into the intermembrane space. Okay, so now we've dealt with our hydrogen ions. Our NAD plus goes back to the Krebs cycle, and our electrons have moved on until they get to protein complex 4. Well, the other reaction, the other electron carrier that we used is FADH2. So he was made in the Krebs cycle. Okay, he is also going to release 2 electrons. Okay, and then he will split into FAD, no pluses, okay, and 2 hydrogen ions. Okay, so this FADH2 splits into 2 electrons, 2 hydrogens, and 1 FAD. Okay, these 2 electrons will also get shuttled into the Q complex. They will then also go to cytochrome C, and then they will also end up in protein complex 4. When this cytochrome C and Q move through the 3rd complex, they're going to take these hydrogens and they're going to pump them over to the other side. Okay, so we're going to end up with hydrogen plus, hydrogen plus. All right. All right, so what are we starting to accumulate here? We're starting to accumulate a lot of hydrogens as this process is repeated, because we made 2 FADH2s, 1 for each pyruvate. We made 4 plus 4 in the Krebs cycle for each of the NADHs, and then you also have the NADHs that were made in glycolysis. Now we need to deal with what's going to happen with these electrons, okay? These electrons are going to use oxygen, okay, plus 4 hydrogen ions, okay? So we've got 2 electrons from the NADH, we've got 2 electrons from the FADH2, so that means we really have 2 sets of electrons. We have 4, okay? And then they are going to come together with these guys, and they're going to make 2 water molecules. So because of this, because this is the end of the line for the electrons, we often refer to these oxygens as being the final electron acceptors, okay? This process, starting from protein complex 1 and ending at protein complex 4, this is the electron transport chain. Okay. as the electrons move down the electron transport chain, we end up with a buildup of hydrogen ions. Okay? Remember that this is a very, very narrow space, so we're quickly coming out of homeostasis. So that's going to trigger our ATP synthase. ATP synthase is going to take some of these hydrogens and it's going to suck them through his little tube to the other side. So this puts hydrogens back over here that can be used to form electron carriers or to be sucked through the inner mitochondrial membrane into the intermembrane space. But when this top part, this transmembrane part of the ATP synthase sucks the hydrogens through, that causes this bulbous part at the bottom to spin or rotate. When he spins or rotates, his job is to take ADP plus a free phosphate ion and make ATP. Okay, so he takes these two guys and he's able to put them together to make ATP. Remember this right here, this is the whole point of doing cellular respiration. Now, just like this part was actually the part that was called the electron transport chain, this part here is called chemiosmosis. Okay, so it is chemical osmosis. It has to do with the movements of water, and it is what is creating the ATP. This is driven by a buildup of hydrogen ions in the intermembrane space. So this whole thing is actually not called the electron transport chain. The electron transport chain plus chemiosmosis is actually called, oxidative phosphorylation, okay? It's called oxidative because oxidation is losing electrons, so all of our electron carriers are losing their electrons, and it's called phosphorylation because we're phosphorylating an ADP. All right, that was a lot of information, so let's see what is important that we need to write at the top, okay? So one of the things that we're going to write at the top is that our NADH and FADH2 molecules carry the electrons to the electron transport chain. We want to make a note that oxygen is the final electron acceptor and will then form water. We want to make a note that the intermembrane space is small, so the hydrogen ion concentration gradient is created quickly. Why do we care about the hydrogen ion concentration gradient? Well, the hydrogen ion concentration gradient activates the ATP synthase. Our starting molecules or reactants are NADH, FADH2, and O2. Our products are ATP and water. All right, so now we're on to fermentation. There are two types of fermentation, so I'm just going to quickly take and divide my fermentation section in half. I'm going to start with alcohol fermentation or alcoholic fermentation. All right, with alcohol or alcoholic fermentation, you're going to start with a glucose molecule. We're just going to use words for this. We're not going to care as much or focus as much on the number of carbons or anything. We're going to actually do glycolysis. So if you remember, we're going to go from glucose and we're going to make 2 pyruvates or pyruvic acids. Okay, and in the process of doing that, we're going to take 2 ATPs and we're going to make a net of 2 ATPs. Remember that in glucose, you're actually making 4 ATPs, but you have to invest 2 at the beginning to get it started. So we're just focusing on this area here. And at the same time, we're going to take 2 NAD pluses and we are going to capture the electrons and make 2 NADHs. And if you remember this process, I'm just going to box out our molecules so that they stand out a little bit more because we're going to add a lot of information here. That this process going from glucose to pyruvate, that this process is called glycolysis. And remember that glycolysis happens in the cytoplasm, so all molecules, all cells can do this, okay? But at this point, we are in a yeast cell, and a yeast cell doesn't have mitochondria. So how can they, what we need to do is be able to regenerate this NAD plus so that we can continue this glycolysis cycle and make that little 2 ATP amount. So we're gonna go from 2-pyruvate to 2 ethanols. And when we go from 2-pyruvate to 2 ethanols, we're going to release a carbon dioxide. So if you remember, glucose was 6 carbons, pyruvate is 3 carbons, and so ethanol is a 2-carbon molecule, but not something that you need to remember. So one of the things that we're going to input here in order to get to this ethanol is we're going to use that NADH that we just made. Okay, I'm going to use another color for that. I got rid of moved my red pen, so I'm going to use pink. So this guy is going to come down. Okay, so what we just made in glycolysis is going to come down. We're going to use him. We're going to input him. He's going to drop his electrons off. So that's going to give us 2 NAD pluses. Okay, and then these NAD pluses are going to go back and be able to use with more glucose to form this 2 ATP in glycolysis. So this process here is called fermentation. We call it fermentation. But really, it is an NAD plus regeneration. That's why we need to do fermentation, or why yeast cells need to do fermentation. They need more of this ATP, and in order to make more of this ATP, we have to be able to regenerate or make this NAD+. We need to free it up so that we can make this 2-ATP. So we're going to write here that this is going to occur in yeast cells. Okay? So that is alcoholic fermentation. So the other type of fermentation is lactic acid fermentation. Lactic acid fermentation is going to start the same way in that we need to start with glycolysis. So again, we're going to start with glucose. We're going to go to pyruvates. In the process of going from glucose to 2-pyruvates, we're going to take 2-ADP. We're going to make a net of 2-ATP, and we're going to make 2 NADHs. So remember, this is just the same glycolysis process, just simplified here, that we talked about on the second flip book, okay, right here, same one. We're just skipping all of these big details, right? All right, so now we are no longer in a yeast cell. So this time we're gonna do lactic acid fermentation. This mainly happens in 3 different types of cells. I'm gonna go ahead and write them down here. This is gonna happen in yogurt making bacteria. This is gonna happen in our red blood cells. And that's because they have no mitochondria. Red blood cells are weird. And then it's also going to happen in our muscle cells. That's typically what we have focused on when we were in 9th grade biology. In muscle cells, it only occurs because muscle cells have lots of mitochondria when there is little to no oxygen. So this would be when you are strenuously working out or running, okay? So we've done the glycolysis, and we have, we're in an organism with no mitochondria, or we have little to no oxygen, okay? So what we're gonna do is we're gonna go from this pyruvate, and we're gonna make 2 lactates. Lactates are also known as lactic acids. Okay, and guess what we're going to use to go from this pyruvate to lactate? We're going to use these 2 NADHs that we just made in glycolysis. We're going to input them, drop off our hydrogen and our electrons, and we're going to make 2 NAD pluses so that they can go up and be used to regenerate more ATP. So this still is fermentation, right? But what's the process? What's the purpose of fermentation? The purpose of fermentation is to regenerate this NAD plus so that glycolysis can occur over and over again. So we do want to remember that this is also an NAD plus regeneration. All right, so while we're here, I do want to take a moment to talk about these products, okay? So in both cases of fermentation, we're getting a product of NAD+, but that's just going to go back into glycolysis, so that's not a product that's released. In yeast cells, we're going to make some alcohol and we are going to make a carbon dioxide. Now, when we are using yeast cells to make bread rise, the rising of the bread is because of this carbon dioxide that is made. Now, the amount of alcohol that is made in bread is very, very little. It is not enough to cause any type of intoxication, and even if the bread is raw, and it will be cooked out when it is cooked. It'll evaporate when it's cooked. Lactate or lactic acid will build up in our muscle cells, and it is toxic, but our muscle cells will shuttle that lactic acid to our liver. That will break it down so that it is not toxic to us. But that is, lactic acid is what causes you to feel the burn, because it's an acid, when you are working out really hard and you cannot physically breathe enough to get enough oxygen in to do regular aerobic respiration. All right, so let's look at what we're going to write at the top. Fermentation itself does not generate ATP, but it does regenerate NAD+, so that glycolysis can continue. Did I spell that right? Yeah. All right. Alcoholic fermentation takes pyruvate and produces CO2 plus ethanol and lactic acid fermentation takes pyruvate and produces lactic acid. All right, that is it. So make sure that you study this foldable, that it is in your notebook, and you have all the key pieces to cellular respiration, whether it is aerobic with oxygen, so that would be glycolysis through the electron transport chain, or whether it is anaerobic, which would be glycolysis plus fermentation. Thank you so much.